Hello, everyone, and thank you for attending the Internet of Waters Peer-to-Peer Network webinar series. Today, we're going to be talking about water data for COVID and wastewater surveillance programs. I'm Ashley Ward. I'm the Engagement and Outreach Associate with the Internet of Water. And I want to go over a few quick housekeeping rules before we get started. Look, there's my name. The first is to remind you that we are recording this webinar, and the presentation slides from today's webinar will be available on our website. And those of you who registered will receive an email with the link to the video as well as the slides. I also want to let you know how you can engage with the presenters during this webinar. Please, if you have problems with the webinar, for example, you can't hear volume or something like that, please put those questions in the chat box, and one of us administrating the webinar will make sure that we get to you quickly. If you have a question for one of the speakers, please place those questions in the Q&A box. For those of you who are participating via WebEx, we're so glad you could join us. You will put all of your comments or questions in your chat box, and one of our administrators will forward that on over to us so that we can get your questions handled. I'm very excited about today's webinar. We have a great group of folks to present to you about the work going on in their states, in Utah, in New York, and in California. And so what we're going to do is I'm going to introduce each of them right before their presentation. And we'll have a Q&A session at the end of the presentations to answer any questions. However, if you have a question specifically to a presentation, go ahead and stick it in the Q&A. And if we have time at the end, I'll hop in and ask the question. So while we're loading up the next, Nathan, I'm going to go ahead and let you start sharing your screen. I'm going to go ahead and introduce our first speaker, Nathan LaCrosse. Nathan is an epidemiologist. Oops, sorry, my screen just went a little berserk. Nathan is an epidemiologist with the COVID Active Response Team at the Utah Department of Health. He received his Master of Public Health in 2006 and his PhD in epidemiology in 2011, both from the University of Michigan. He joined the Utah Department of Health in 2013. Working with environmental epidemiology on environmental health issues. And since March of 2020, he's been working on COVID-19 surveillance and assisting the state's pandemic response. In November, Nathan moved to a new position within the Utah Department of Health to take a larger role in Utah's Wastewater Surveillance program, which is what he's here to talk with us about today. So Nathan, welcome.
Thank you, Ashley. So as Ashley mentioned, I'm more than happy to talk some about our wastewater surveillance program here in Utah. We've had it going for a number of months now. See if we can actually advance my screen. There we go. So real briefly first, some basics about wastewater surveillance. A lot of it is as you would imagine it is. If you imagine a sewer system, and there's a very simplified graphic hopefully on your screen, somebody please tell me if it's not, you're collecting sewage, wastewater, from a pretty wide area typically, although obviously that's going to vary by the community in question. There's going to be, a number of houses, businesses, stores, etc. In this case, we're kind of just showing homes. In the current pandemic, some of those homes will contain people that are currently infected and they are shedding the virus in their feces, because this is one of the viruses that can be shed in that manner. That virus will flow through the wastewater system and then ultimately get to the wastewater treatment plant. That is the point where typically we're going to be taking a sample, the point at which that wastewater stream enters the plant before treatment. And then the rest of the wastewater will undergo the normal treatment processes and then be discharged. And I've heard some anecdotal stories of genetic material, which is not to necessarily indicate intact viruses at all. We're not even sure that there's really much, if any, viable virus in untreated wastewater. But typically you don't really find anything in the treated wastewater that gets discharged, particularly nothing viable. That's one of the main purposes of wastewater treatment plants, is in fact to get rid of that sort of thing. Once you have your sample, there's a number of different ways to analyze it. There hasn't been sort of the one true method hasn't really emerged at this point. There's often some sort of concentration step, often involving centrifugation, because you want to typically separate the liquid supernatant from the more solid material, of which there may be quite a bit. And then there's going to be some sort of extraction and capture method, often involving binding to a filter of some sort. Like I said, there's a few different ways with varying levels of efficiency and efficacy and cost as well. But once you have purified viral RNA, the rest of the process is really, in most ways, largely identical to how clinical samples are processed. So these are like throat swabs, nasopharyngeal swabs. They go through real-time quantitative PCR to quantify the amount of viral RNA that is present. And then you have to, of course, express your results in some fashion. For clinical tests, that's often just a kind of a yes/no, whether it's present or not. We actually want the actual quantity. So again, there's a variety of ways to do this. What we ultimately settled on here in Utah is to express the results in the number of gene copies per 100,000 people within that sewer shed per day. So normalizing it to the actual population that's present as well. And then we're also standardizing it by the amount of sewage flow that goes through past that sampling point, because that can have a pretty major impact on the amount of RNA you ultimately detect as well. And one of the things, like I mentioned, there's a number of different ways to do this. I think at least at this stage, until possibly one sort of the best method is determined, one of the most important things is to keep your methods consistent, because you want to be able to compare that data. If you keep changing methods, it becomes really hard to tell whether any differences you are finding are due to actual differences in the amount of viral RNA in the wastewater, or if it's just due to the fact you keep changing your methods. Okay, real briefly about Utah's program. So it's there's a lot of different moving pieces, and we're kind of in the midst. We'll just kind of coming through some recent changes. So we're partnering, of course, with a number of different wastewater treatment facilities, and they're the ones who are actually collecting the samples, because of course they have access and the equipment and familiarity to do so, and they're filling out some chain of custody forms for us with some important info. These get transported to laboratories. We currently have 4 partner laboratories, and these are academic labs. We're looking at bringing our public health lab online, hopefully in the coming months. Some of them are also participating and helping out with transport analyses, because we have a reasonably large state in Utah area-wise. They're of course doing the sample analyses and helping develop and refine our analytical methods. And then here at the Department of Health, we're sort of doing the coordination. We're doing some of the sample transport as well, data management, trying to figure out what does the data actually mean, and then communicating it out and making recommendations as necessary based on it to CDC, local health departments, other folks within the state health department, policymakers, anybody who could potentially make use of the data. So kind of here's what our project currently looks like. Of course, ultimately the goal of all of this work is to support our state's response to the COVID-19 pandemic. In a variety of different ways. Currently, we're sampling at 33 different facilities. The ones without the red X's, we had been sampling at 46 previous to this month, January. What changed is we moved from sampling once a week to twice a week in an attempt to make the data more timely and more easily actionable and more useful. But of course, that is easily more than twice the effort and twice the resources you need, especially for some of these remote areas. And so we couldn't keep sampling at all of these sites. And some of the facilities couldn't keep sampling twice a week as opposed to once a week. It was just too much burden for some of the really small ones. So we ended up having to cut some, but we're still sampling about 87% of our population. And that's largely because most of the population lives along the Wasatch Front, along these mountains. And that's that sort of line of dots that kind of curves along the north central portion of the state. That's where Salt Lake City is, where Provo is, where Ogden is. It's where a large majority of the population lives in Utah. We're presenting it out in a few different ways. We have a publicly facing dashboard that's been up since the summer, and it's undergone a few revisions and changes as we kind of refine things. This is kind of how it will look in the very near future. It's not too dissimilar currently. So there's basically a statewide map, which is the map I showed you previously, actually. You can click any of the dots and it'll kind of zoom you into that particular wastewater facility and then display in the top graph the actual wastewater surveillance copies per person per day, as well as the daily case rate, so the number of cases per 100,000 people within that same sewer shed per day, so we can start comparing some of these data. The data are also presented on a much more restricted access internal dashboard for the Department of Health and our unified command and some other public health and policymakers. There's not much difference currently, but they may diverge further in the future. So I'm going to show some example data in a minute, but I always find it helpful to kind of put interpretation and context to think about some of the limitations on this type of data. And one of the main limitations is that the data can often be variable. And a lot of this is because there are a lot of potential sources of variability, and we don't really have a great understanding necessarily of what all these sources are, how they impact the data, and how best to account for them. So there are things like differences in the sewer system infrastructure. I suspect it's similar in other states, but certainly in Utah, the age and the type of treatment plant and what kind of sample they can take and lots of other factors can differ pretty significantly from facility to facility. You can have varying amounts of infiltration of runoff and groundwater, especially some of the old facilities are really leaky. And so in the spring, quite a bit of their flow can be comprised of this runoff and groundwater. And that's going to change the dilution certainly, but also how long it takes for the sewage to go from for example, a home to the sampling point, which is the next item here, the wastewater transit time. And that has implications for viral RNA degradation because this does degrade over time. And so in theory, the quicker you move it from point A to point B, the less degradation that will have occurred. Temperature can play a role, especially in that degradation process. It occurs significantly faster at higher temperatures. There often are other constituents of the wastewater stream, often from some of these industrial sources. These can be things like detergents, soaps that can increase degradation of RNA, probably other chemicals that can have unknown effects. And it can be really hard to find information about even just overall how much of the wastewater flow is due to influx of industrial sources, much less what is in those industrial wastewater streams. And there could be biological differences too. So which people or populations tend to shed the virus? It may well not be everybody, at least not all the time or throughout the course of their infection. How much virus is shed? That does differ for people. We don't necessarily have a great sense for exactly how over larger populations, but there's increasing data coming out in the literature that shows that it can certainly vary quite significantly. Similarly, how long the virus is shed throughout the course of infection can vary significantly from person to person, and there are likely to be populations that differ on these factors, and the makeup of the different populations in various sewer sheds can also differ. So when you take all these different sources of variability together, what it really comes down to is it's really, in my mind, not feasible to compare data very easily between different facilities. Right now, there's just too many differences to really get at what do the differences mean? Is it real differences in the amount of basically the number of people shedding the virus upstream, because that's really what you want to know, or is it just because there's all of these other differences within the facilities and within the populations that are influencing the data? So we don't do that. What we do is take data over time within a facility and look at the data there. We're looking for trends typically within the data from a single facility. And we're doing this over, currently 33 different facilities, but we don't compare the absolute magnitude from facility A to facility B, because I don't really think they're comparable right now. And then, like with most other surveillance data, it's best to interpret this whenever possible with whatever other information you may have about the population in question, what's going on in that area, because this can have pretty big impacts on what you see. And I'll show a little bit about that. So data, I think one of the main and most useful uses for this data is really to serve as a leading indicator of rising infection rates, which it can do very well. We have really, with the current number of cases, we're having all too many examples of this. So this is example from the city of Provo from back this previous fall. You can see again, the top graph is the actual wastewater data. The bottom graph are the daily case rates within that same area. And we can see, it doesn't look like much now because the y-axis ended up going much, much higher. But right here, August 18th was the first day that kind of triggered my eye when I was looking at this data and figuring out, is this important enough to inform our local health departments and other public health leaders to, hey, there's might be something going on in this area. And here is where it caught my eye. And this is actually considerably more of a lead time than we often get. We also ultimately had rising case rates that went up much higher than we're seeing during the summer. And this was, of course, reflected in the wastewater data as well. Typically, we'll often see about maybe 5 to 7 days of lead time. So often we'll get an increase in the wastewater data, maybe 5 to 7 days. It really depends on the facility and the population and the time in question. 5 to 7 days later, we'll see a corresponding increase in the cases identified. That, of course, is subject in a large part to the actual clinical testing that's going on in that area, because that's how I identify the actual cases, through clinical testing. So if there's a lot of clinical testing and the data reporting is really good and it's getting into your database really soon, then maybe that lead time is less. If there's not much clinical testing going on, or it takes longer, more days for that data to actually get reported and be available for comparison, then maybe your lead time is a little bit longer. So to some extent, it's not solely biological, the differences we see. There's a lot of excuse me, public health and programmatic factors that really go into the lead time that we see. But we do often see it, and it can be very useful. Even just a few days at hand can make a big difference. It can also be helpful in sort of confirming declining case rates. So this is something that came up, not something recently, unfortunately, but the end of that past surge we had in June and July. We saw, this is data for Salt Lake City. At the top are the number of people, unique people tested every day throughout the same time period. Then we have the middle graph, wastewater data, the bottom graph, daily case rate in the same area. This is for the entire county, but it's similar within this sewer shed. This is the northern part of the county. We can see that we had nicely declining case rates throughout July, which was great news. However, we also saw declining numbers of people being tested. So there's always the worry that you're only seeing fewer cases because you're just not testing as many people. The cases are there, you're just not identifying them. We were able to lend some confidence to the idea that this decline in case rates was probably real, because we saw a very steep and consistent decline in the wastewater data as well, which suggests that there weren't as many people shedding the virus within that sewer shed. It could be very helpful to provide data where other sources of information like clinical testing is hard to come by. So especially in some of our smaller rural communities, Blanding City is one of those. There's often, the availability of clinical testing is not what it is in some of our more urban centers. And so it can be difficult for public health personnel to get a nice sense for what is actually happening out there. Wastewater data, if you're able to get the data, and it is considerably more expensive and difficult to get samples from these more remote areas, but if you can do it, it can really provide some very helpful information that may not be otherwise available from other sources. And then you will end up at some point capturing some of these sort of transient events, whether they're travel tourism, whether they're state fairs or county fairs, whether it's the big rally or a concert or something like that. It's important to remember when you're looking at this data, we think about it often in terms of sampling the people who live within a sewer shed, but that's not really the case. What we're doing, we're sampling anybody using the bathroom at that time. They don't have to live there. They could be traveling through, and this is what we see in places like Moab, not a big city, about 5,000 or so. There's about 10,000 are being sampled throughout the entire sewer shed. But it is a big hub for recreation and travel in the summer. And we see some of that, especially in this really high peak we saw just a single week back in September. That was not reflected at all in the case counts. We saw something kind of similar back in May and something similar around the beginning of July or so. Around Memorial Day-ish, around the 4th of July-ish, not really a holiday so much. I kind of interpreted it as sort of the end of summer, people taking a last sort of vacation. But we've seen this in a number of different areas where we will occasionally see spikes, and they do tend to be very short-term in duration, that don't ultimately end up being reflected in the daily case counts. These are often in areas where we would expect increased travel and tourism. That, you know, we kind of know these places. And this is what I was getting at when I'm saying that it's important to take other information you may have available into account, like that an area has a lot of people moving through because it's a big hub for recreation. Because that is important information to have in the back of your mind as you're looking at this data and trying to figure out what does it mean, and should I recommend some sort of public health action be taken based on this data. So some other things that we're using it for, I kind of mentioned the first one, of course, identification of high infection rate areas. Typically the response to that would be to send out increased, or basically allocate increased clinical testing resources to that area. We're doing that extensively currently. We're kind of having this testing surge where we're sending out mobile test teams to 30 some sites a week, twice a week, and wastewater data is one of the sources of information we're using to help pick those sites. It could be used for contact tracing prioritization. I don't know if we ever actually implemented this, but it was certainly a subject of discussion for a while as our case counts increased and our contact tracers and other public health personnel were getting overwhelmed by the demand. If it comes to the point where you can't do adequate investigations and contact tracing on everybody, you have to come up with a way to prioritize who you are going to do. Wastewater data is one source of information you can use to do that prioritization. You can use it to assist the interpretation of other data. I kind of mentioned some of that. Where to send mask wearing observers. So we send out people to look around and get a sense for how many people in an area or at a specific location are actually being compliant with mask wearing orders, to get a sense for like how effective are our campaigns to increase that. And you can use it, of course, to target outreach, education, other interventions. So I mentioned public dashboard. We have an internal dashboard. The last thing that we do is I write up, every time we have new data, I'm running up a very brief summary. You can kind of see a little snippet of it right there. Very brief summary of the most recent data, the date, and kind of basically how I'm categorizing the data. So whether it's increasing, whether it's high, whether it's low or decreasing, or whether it's kind of indeterminate. Like it looks like there may be something going on, but it's a little too early to tell exactly what yet. And I do this because, you know, everybody in this response is incredibly busy, and you can't just dump a new pile of data on them and say, this is really useful, figure out what to do with it. So making it as easily digestible and as easily used for the people who can use it as possible via something like a summary report. This isn't necessarily the best way, but it's a way that I came up with, I think is a really important thing to do. Very briefly, I suspect I'm running short on time. A few lessons that we've kind of learned. We had a pilot study early on with a very limited number of sites, I think 10, and that was very useful. It allowed all of the logistic, well, not all, a lot of logistic problems, methodology problems, analytical problems to kind of be resolved before we really expanded it out. And it would have been much more complicated to try to deal with those issues at that point. So as I mentioned, there's a lot of moving pieces. There's a lot of different agencies and facilities and labs and universities that are involved. And so good collaboration, good communication, good coordination are all incredibly important. You have to be constantly communicating and making sure that everybody's on the same page and they all know what their roles are and what they need to do. So I mentioned that there's a lot of different sources of variability. That is absolutely true. I think this field is changing constantly. It's pretty early on in it, but despite the fact that we still have a lot to learn about it, it can and does already provide some, I think, really useful information for public health efforts. And a lot of times information that's not really available from other sources. Water data is often available first. It's important, of course, to select your sampling and analytical methods with public health uses in mind, if that is the ultimate purpose for the data. Like looking back on things, like we started this back in March, like we had no idea what we were doing, really. We were kind of figuring out as we went. Looking back on it, I think we would have probably started sampling twice a week right from the very beginning, because it ended up being pretty clear over the course of the summer that to really make the data as usable as possible, you want it to be as near real time as possible, as one example there. And then, as I mentioned, this is rapidly changing field. You got to be adaptable. It's going to change under your feet tomorrow, but that's okay. It just means that we're learning and we're constantly getting better. So thank you. I'd be happy to take any questions you may have. And visit our public dashboard. It's cool.
Thanks, Nathan. So there is a question in the Q&A, but it's more of a general question. So I'm going to hold that to the end of the session. And let's get Joe, if you want to get your slides loaded up while I intro you. So our next speaker is Joe Fiegel, and the head of Erie County Division of Sewer Management, which is responsible for the 7 Erie County sewer districts in New York. The Erie County sewer districts include 6 water resource recovery facilities, almost 100 pumping stations, approximately 1,100 miles of sewer and other appurtenances. The Division of Sewerage Management is staffed by around 230 professionals dedicated to this operation, maintenance, and management and administration of this infrastructure to protect public health and the environment and the areas surrounding the city of Buffalo, New York. Prior to joining Erie County, Joe worked as an engineering consultant. He's a licensed professional engineer with New York State, a board-certified environmental engineer, and a graduate of Northwestern University. And so welcome, Joe, and thank you for talking to us about your programs there in New York.
Thank you, Ashley. I appreciate it, and thank you to everyone participating in this webinar. I'm here today to offer up a bit of a local perspective on the topic of wastewater-based epidemiology, and also from the perspective of a utility that's responsible for the wastewater infrastructure. Erie County wasn't necessarily selected for this webinar because we have the largest program in the country or because we are the furthest along. As was noted in the webinar description, the peer-to-peer network wants to foster real conversations about successes and challenges, talk about projects in progress, lessons learned, et cetera, et cetera. I'm sure and I'm hopeful there are some watching today that will be able to identify with our story and hopefully learn from our process. So before I venture into the technical details of our program, I thought it would be helpful to provide some background. Erie County is located in Western New York. As was mentioned in my bio, our most recognizable municipality in Erie County is the city of Buffalo, but there are over 40 towns, villages, and cities within the county. All told, Erie County has a total population of approximately 920,000 people. While describing what we have referred to as our wastewater-based epidemiology program, I think it's important to talk a little bit about our governance structure. New York is what's referred to as a home rule state. There's been a lot of discussion regarding states' rights on the federal level and on the federal level. In New York, many rights are provided to smaller levels of government. Per the New York State Constitution, counties, cities, towns, villages are general purpose units of local government with broad powers to provide direct services to the area within their boundaries. Some say that the home rule rights in New York are some of the most far-reaching in the nation. And what that means is there's an extremely decentralized structure to many local services. One of the results of New York's home rule setup is that locally there are more than 20 governmental entities within Erie County alone that provide some form of sanitary sewer service. It actually used to be worse. Over the course of time, the Erie County sewer districts have completed a number of mergers whereby a local government transferred their systems to the county through very laborious procedures due to New York State home rule. To be fair, there are many arrangements where there are cooperative agreements in which one municipality manages the local sewers and the flows collected are transmitted to a larger facility owned, operated, maintained by another municipality. Nevertheless, it's a very, very decentralized service structure. So what does that have to do with COVID-19? Fortunately, with regard to emergency response, a single entity, the county, takes the lead locally. Specifically for this event, it has been our Department of Health, along with our Department of Homeland Security and Emergency Services at the county level. As with many locations throughout the world, Erie County has been severely impacted by COVID-19. One of many metrics being tracked by the county's health department is the number of hospitalizations and intensive care unit patients. The data presented here provide context for the manner in which our community has been affected by the pandemic. Our first surge in infections occurred during the spring. By summertime, infections had decreased substantially. However, by the fall and to the present, infections and hence hospitalizations have surged again. A critical component of Erie County's response has been management of large volumes of data. GIS has been one tool used. Erie County has a fairly robust GIS infrastructure that is used in many applications. As an example, with the county sewer districts, GIS is a central component of our asset management initiatives. During most emergency events, the county's GIS section is a critical resource that is almost always mobilized immediately. With the current pandemic, a number of different metrics are managed and/or displayed through GIS. Pictured here is a screenshot of the county's public-facing dashboard. This portion of the dashboard presents confirmed positive cases by zip code. Zip code specific data has been a major way the county's Department of Health has been performing their epidemiology tasks and COVID response. Much of the data collected by the county are reactive, i.e. metrics such as hospitalizations, patients in the ICU, and the like are collected after people have gotten really sick. During the summer, when the COVID cases were lower locally, Erie County began discussions on implementing a program to test wastewater for the presence of the SARS-CoV-2 virus as a potential tool to assist with the community's response, as it was seen that these data could provide a type of early warning system. As Nathan described, I thought very well, COVID-19 infected individuals shed the SARS-CoV-2 virus in their stool. Within a sewer system, a known volume of wastewater may be collected and processed to determine how many virus gene copies are present. In simplistic terms, the more gene copies per liter, the higher concentration of community spread in the area of the sewer system upstream of the sampling location. It was our hope through this program that we'd be able to provide additional information to assist the epidemiology section within our health department to make better informed decisions. There's some research that a rise in the concentration of gene copies in the wastewater may precede a rise in hospitalizations by a week. That could provide local officials valuable lead time in the midst of a pandemic and would be different than some of the more reactive data collected. After researching the matter, the county coordinated with the University of Buffalo Engineering School, which had equipment and expertise to perform the qPCR laboratory work on the wastewater samples. And then we set forth to develop a pilot program. One of the first challenges was to determine where to sample. There are literally thousands and thousands of miles of sewer within Erie County, and the pilot was to include 10 sampling locations. A decision was made up front to gather 24-hour composite samples, which were viewed as more representative since a sample is taken over a full day as opposed to a single grab. One challenge is that 24-hour composite samples require equipment that is in short supply. Another problem to overcome harkens back to the governance discussions earlier. There's an extremely decentralized service structure that presents issues regarding coordination and other factors. So how did we get it done? Well, when scoping the project, we wanted to make sure we could maximize the coverage for the 10 samples and minimize the complexity of the sampling effort, while also providing a variety of areas covered to test the efficacy of our program. After reviewing existing sewer sheds, we were able to boil down the 20+ municipalities into 4 that manage the 6 largest wastewater treatment plants in the county. At each of these treatment plants, the municipalities have existing systems to gather the necessary wastewater volume over a 24-hour period. This strategy allowed for the majority of the major populated areas within the county to be captured. One negative was that in certain instances, large geographic areas would be covered by a single sampling location. For example, the largest facility services a population of over 400,000 people. Fortunately, most were much smaller than that. After review, it was determined there were no other good alternatives, as with thousands of miles of sewer and a limited supply of sampling equipment, and then coupled with the uncertain nature of where the next COVID-19 outbreak may occur, there was the proverbial needle in the haystack effect when deploying samplers to other remote areas. There was, however, one exception. The start of the pilot coincided with the beginning of the fall semester for the University at Buffalo. Understanding the upcoming changes in the population characteristics and the higher potential for variation in COVID-19 infections when compared to other areas in the county, the school's North Campus was specifically targeted with the deployment of portable 24-hour composite samplers to capture both on-campus and off-campus housing around the University of Buffalo. Samples were taken twice per week at each site. Personnel from the University of Buffalo picked up the samples from the 4 participating communities on Wednesday and Friday mornings and transported them to the laboratory. The days were selected to coincide with a similar pilot being performed by New York State in conjunction with Syracuse University. Working with the state's program was helpful as we were able to learn about their methods and draw from the expertise at Syracuse University as well. There were 2 shared locations in which split samples were provided for both New York State and Erie County programs. As mentioned previously, the summer period in Erie County coincided with a time of lower hospitalizations and lower community spread of COVID-19. The sampling results bore that out. Most of the samples collected during the pilot period were below the limit of detection. The samples that had the highest spike were indeed those associated with the University of Buffalo North Campus. The data in blue on this figure captures the entire campus and the major off-campus housing area. Reports from the County Health Department indicated the rise did indeed coincide with increases in infections on the campus, and there were some indications that an early response was found in the wastewater data. The pilot was certainly promising, and as a result, we chose to continue sampling while a decision was made on funding for additional testing into the future. The University of Buffalo would preserve the samples, so if or when funding was identified, we had a full complement of data over the entire time period. In October, we received good news that that additional funding was secured to further the program. The partner municipalities continued to collect the wastewater samples for storage and preservation. The University of Buffalo proceeded to order the necessary testing kits. This is where our program hit its most significant challenge. There were major supply side issues with obtaining the testing materials for our program due to demand and production limitations. The laboratory's preferred vendor temporarily stopped making environmental kits so they could produce more for the community COVID testing. This went on for several weeks. The University of Buffalo tested RNA extraction kits obtained from other vendors, but found those kits did not work quite as well as those of their preferred method. Alternate kits did generate some results, and in the meantime, the laboratory was working to increase sensitivity and recovery in the samples, while hoping the supply of the preferred kit would shake loose. In December, the University of Buffalo finally worked out the supply chain problems with their vendor and obtained the kits to analyze remaining samples. Very recently, and when I mean by recently, I mean within the last 2 days, we started to receive the data. The data presented on this slide are very preliminary, but some of the observations include the fact that our program has very good sensitivity and high detection, and that we're able to detect SARS-CoV-2 in locations that have very low reported cases, which is a really good sign. The wastewater samples reflect trends seen in prevalence and community transmission, with copy numbers increasing exponentially as cases increase. The third observation that we made, again, just looking at this data over the last couple of days, is preliminarily, it does not appear that there's a strong early detection trend with many of our sites. One of the theories that we have is because the COVID outbreak is so widespread and many of our sampling locations cover large sewer sheds, the level of resolution is really not available. This is an item that we're going to be analyzing further over the next couple of days and into the next few weeks. One thought is once community spread is less prevalent, better resolution will occur, particularly at the sampling locations with smaller population or those that are closer to the source. Moving forward, we're hoping to have data within 24 hours and share results directly with the Department of Health. We are considering developing a dashboard that can be updated in real time. We're also looking into presenting the data based on zip code, perhaps through a GIS platform. As referenced earlier, managing data by zip code is a primary way the Department of Health is tracking pandemic metrics. Unfortunately, sewer sheds do not follow zip code boundaries, but nevertheless, in conjunction with the University of Buffalo, we want to provide these data in forms that are best for the epidemiology staff. The engineer in me loves this as a scientific experiment, but ultimately everyone involved wants this to be as useful a tool as it can be for the folks on the front lines battling COVID-19. With that, I'd like to thank everyone for your time, and certainly will entertain any questions that may have come in.
Thanks, Joe. There are a couple of questions, but I think like before, I'm going to hold them to the end because I think all of you could probably contribute. So while you are wrapping up or shutting down your computer, your sharing, let me introduce Greg Gearhart, our third presenter today. Greg is a longtime friend of the Internet of Water and a wonderful partner in this network, the peer-to-peer network, and bringing together folks for conversations like this one. And in fact, was initiated by Greg. He is the Deputy Director at the California State Water Resources Control Board and is a passionate advocate for data modernization efforts and that open and democratized data for improved water management decisions. He's been the director of the Office of Information Management and Analysis for 5 years. Prior to that, served as the statewide storm water program manager, as well as in many different program areas, including wetlands, watershed management, organizational development, and enforcement. Greg received his degree in environmental resources engineering from Humboldt State University. So thank you so much, Greg, for talking to us about the work in California.
Thanks for having me, and great presentations by Joe and Nathan. Thank you for preceding and setting the stage. I would like to hearken upon the Monty Python line of now for something completely different. Full disclaimer, I am not a public health person, so you will not be hearing me discuss the implications of any results and data that has been coming in California, but I've also been part of an honored to be part of an amazing team in California. There's a lot happening in California. It's not surprising to many, perhaps, that California in many ways acts like its own country. And so the number of projects and activities and data collection to date is actually unknown. So it's kind of interesting. We've been doing, I think in California, sewerage monitoring since the beginning of the epidemic and pandemic, but we don't have a single collection data pipeline yet. And that's actually what I'm going to talk about is some of the data challenges. That's what I know the best and what I really hope the Internet of Water and this peer-to-peer network can focus on. So we're also sort of collecting anecdotally at this point, interpretation and use of the data. So we're not actually in a position, both as a state agency in charge of water, as well as our partners at the public health, to know everything that's being done with use of this data. There's quite possibly some really good success stories on regional and local scale. There's also a new interest to sort of look at rising incidence because of, or rising prevalence because of the incidence rates and the new hospitalization numbers. So we're in this sort of emerging, fast moving space in California. And we're trying to, my team is really trying to support this effort by building out a data ecosystem that will help support all the different users groups and all the different interests that are associated with it. One of the big things I'll highlight now, and I'll talk a little bit at the very end of my presentation, is the data needs really focus around sort of making the data fair, findable, accessible, interoperable, and reusable, but also the metadata component that sort of makes that sort of happen. So there's two sort of things that happen when you talk about the data that way. And one is the structure of the data so that it's more usable and interoperable, but also just the accessibility implies that you know how to use the data. So context and data quality and all those issues that go along with it. We really need to figure out some standards that would be used across the country and across the world really for informing metadata, which would then inform the users of the data. One of the things that happened in California early on was a need and a desire to sort of centralize some of this data collection. So I want to honor the pilot members that I've become a team member on in California. So in California, there is a CDC grant funded effort through the California Department of Public Health. The water boards have become partners in it. The leadership on both ends has been sort of Mark Starr at CDPH and Karen Mogus at the water board, and lots of other people at a higher level. But those are the two sort of point people. And then I want to just acknowledge also the great staff leadership by Duke Vugia at CDPH, as well as Claire Wagoner at the California Water Boards. I have a team member here on the panel today that can answer any questions that might come up about data, but we've also had a lot of data component progress, and we hope to sort of make more progress in the near future. That pilot project in California is focusing on 5 collection areas. So we actually had, because of direct potable reuse efforts in California already underway, we had some some wastewater folks that were looking at the viability of using that water as potential drinking water supplies. And so they were already sort of looking at genetic makeup in some of the wastewater influent streams. So they had been doing some work similar, and we quickly pivoted early on to get them to look at COVID. Those 5 collection agencies represent large urban areas. So San Diego area, LA County, LA City, Orange County, San Diego or Sanitation District, as well as San Francisco Public Utility Commission. We also have plans to expand this to other treatment works in California, as well as sewer sheds. And so we're considering trying to figure out how to pivot the pilot to also include some of these high priority vulnerable communities that include hospitals and correction and rehab facilities in California. We are at the very beginning of this, so I want to just lay out some of the principles from the data perspective. Again, I know there's lots of great venues out there to talk about the public health implications, and I'm super glad that the prior 2 speakers have expertise and some knowledge in that area. I am going to talk about the data though. And so like one of the key things is that we're keeping our eye on the prize of a CDC, the Center for Disease Control endpoint. So our data is headed for that CDC database in sort of the most general sense. We know though that there's lots of data that's being collected in California that is not destined to go to that endpoint. So, but we want to make sure that we're inclusive of it in our warehouse approach, so that we can provide this data to other people that may be interested, as well as provide the context. And that includes building out some of these components like FAIR, findable, accessible, interoperable, and reusable data. If you haven't looked that up, it's a really interesting set of resources that guide a lot of our work at the Internet of Water in California. We also want to make sure that these data ecosystem principles include being flexible enough to include all sorts of data. So these different methods that might be used, different sewer sheds, and different sort of contexts. And so we want to make sure that that data has a place, as well as the context is available, so that people know how to use it and can easily sort of filter on the kinds of data sets that they want to use. So this ecosystem will include some interfaces that are on the front end and back end, quote unquote, of this warehouse. The back end would be allowing us to transform that data into not only the CDC endpoint kind of a structure, but as well as providing public accessibility. So we envision open data for all the data that's available for the public, and we envision sort of a standardized approach so that all that data becomes accessible without someone having to go and look through different spreadsheets and download and transform their own data. So on the front end, we also envision, because of the different pipelines and workflows, we envision data coming in different ways at different times. And so we're trying to build a very flexible system that allows that front end to also do as much of the work of transforming and quality control as possible. One of the things I want to acknowledge is that there has been a tremendous amount of leadership already on this, not by me or not by my agency necessarily. So local agencies that have been doing this work on their own should be acknowledged. So I mentioned LA County and the Bay Area collections folks. I really want to acknowledge also that in the Bay Area, there is a couple of academic supported efforts. One is through the UC Berkeley approach and the San Francisco Estuary Institute. They're doing a lot of work to collect that data and make it usable and provide a dashboard for the local folks in the Bay Area. There's also a team at Stanford that's doing a lot of really innovative and leadership development oriented work on the implications as well as the methods. Where the Water Board and my team fits in is really in this data side. We're kind of agnostic on a lot of those different issues. We do have part of our team, a QA chemist that is helping sort of guide how to put the context into the metadata, but we're really focused mostly on making the data as accessible and as available in a timely manner as possible. And that really gets to what I think are the key data issues that we face both in California as well as across the country. And one of my goals with the Internet of Water is that we see the growing of a peer-to-peer network. It doesn't have to be big, but it certainly will be vibrant, I bet, discussing some of these data issues. So metadata is really the easiest way to say it. And I know that people fall asleep on the first time I say metadata. That's why I said it 3 times, is to keep you awake. But one of the things that's important is this, not only developing the standards for how to express that metadata and collect it and therefore make it available, but what are the key elements? What kinds of things do we need to make context really important? You heard Nathan and Joe talk about some of the issues that have come up in their state efforts to communicate this information. There's some great questions in the queue that I'm going to stop talking and listen to in a minute. But tourism, like pulses, small sewer sheds. You've heard stories of dormitories doing this. So all of that context has to be somehow communicated effectively. And then really one of the most important things you heard was the importance of quality. Excluding other data from other methods is not always an option. So it might be really important and viable for us to provide some sort of data quality context in the metadata that allows usability across some of these methods. In California, this is particularly the case. As you know, we have a lot of different competing methods and companies that are trying to provide data in different forms. And so this really goes to the next key element, which is that what I talked about, data structure. So we really need to develop some common data models, in my opinion. This applies to mostly the combination of the wastewater data and the sewer shed data and the health data. So some of this is geospatial, some of this is going to be tabular data coming in at different times sequences. And so we really need to sort of tie this all together with health and community data on a time spatial scale that allows this data model to be universally used across the state of California and therefore across the country and maybe across the whole planet. And we have our eye on the prize of not just COVID, but future epidemics and outbreaks that could be detected using wastewater surveillance data. We're really excited about the platform and the data ecosystem. We want to really just build it out so that it's most accessible and provides that sort of findable, accessible, interoperable, and reusable standard. So I will pause and allow us to go to some questions, Ashley.
Thanks, Greg. There are a couple of questions, and before we get to the Q&A, one quick thing I wanted to piggyback off of what Greg raised is we're hoping this will be the first of a series of webinars about this topic, and the second of the series focused more specifically on data issues, data infrastructure, and some of these topics that Greg has raised. And so we hope that you all will join us for that second webinar and at the date to be announced. And so with that, I'm going to switch over to a quick Q&A with Nathan, Joe, and Greg and Rafa. So let me get to the first question, which is about sewer collection systems that are exfiltrating raw sewage into drinking water aquifers, like what's happening in Morro Bay, California. So I don't just open that up for any of you to respond to that question.
Well, we don't draw our drinking water, at least locally here. We don't draw from an aquifer. Of course, we have Lake Erie right in our backyard. It's why we're called Erie County. If the concern is with regard to the SARS-CoV-2 virus making its way into the aquifers, I guess what I can, and Nathan may have some better information with regard to this, but from what I understand, there really, when we first got into the pandemic, our employees were really concerned about the virus transmitting during their day-to-day operations, and the CDC came out and said that there was no COVID specific precautions that need to be taken. And part of that reason is because wastewater and sewage inherently has viruses and bacteria and that type of vectors in it. And so with regard to those protections, there was nothing that was further needed. My understanding is that the virus is not very viable in sewage. So when we are testing for it in the laboratory, we're actually going for fragments of the virus, the RNA of the virus. I don't believe that it is readily available or readily viable outside in the sewage itself.
That's my understanding as well. I wasn't aware that there were areas that were pumping raw sewage into their aquifers. I would also add that, it's viruses are adapted to entering and replicating in living things. And there are obviously ones that can live in bacteria, but this isn't one of those. I wouldn't expect, even if there was viable virus that's being pushed into the aquifer, I wouldn't expect it to remain viable for very long. So I don't know that there's an increased risk of people being exposed to it from drinking water from that aquifer. Certainly not more than there would be from other pathogens that could be in sewage.
So quickly, there was another question in the chat, and I just wanted to raise that as well. Actually, it's a multi-part question. The first, how have your respective state and local health officials responded to your wastewater testing? And the second part of that is, what particular data have they been requesting? So how have your state and local health officials responded to the testing, and what particular data have they requested from the testing?
So at least here in Utah, variable, but overall positive. I don't think anybody's been against it. It's ranged from people viewing it, personally, of course, I think rightly, as a very valuable new source of data to incorporate into their decision-making process, to some various degrees of apathy. I think that for some, especially some of the smaller agencies that maybe don't have as many resources to devote to kind of figuring out and how to think about new sources of data, there's more of an inertia. Really, and it's not specific to this, I don't think necessarily. I think that's probably true of just about any new source of information. A little bit more of a wait and see approach, but in general, it's been quite positive, I think. We've had some of our local health departments ask us to start sampling at additional communities, especially mostly communities where they don't have some of that information that they would really like to see. Smaller ones that don't have a great deal in the way of clinical testing going on, and so they don't really know what's happening out there. And they viewed this, and again, rightly, I believe, as a way to get some of that information. And when we've been able to accommodate that, we've been more than happy to do so. Others have asked for more sampling at a smaller scale. So this kind of gets to the point I think that Joe brought up, I think is really pertinent as well, that there's a balance, and it's a hard one to figure out where the best point in this continuum is between, if you're sampling at the smaller scale geographically you get to, the easier it is to figure out what you should do about the data that you get. It's also a lot harder, I think, to get good data from some of these really small scale areas. And of course, you're spending really the equivalent amount of resources to sample fewer and fewer people. So some of the efficiency advantage that you're getting from this data kind of starts to go away a little bit. As an example, a couple of our facilities in Salt Lake County are very large. They'll cover, one of them covers close to half the county and a little over 500,000 people, which for Utah is really big. I know there are other places where that's not much at all. And so it's, from a population perspective, it's great because you're sampling 500,000 people for pretty cheaply, really. But when we find something and then we convey that to the local health department and they're trying to figure out, okay, what should we do and where? Because there are a number of individual communities, it's this big metropolis area, but there are a number of cities that are all right next to each other within there. The cases aren't going to be evenly distributed throughout that sewershed. We know that. And so for them, it's harder to take action on data from some of these really big populated areas because you're not going to be able to send like mobile testing teams to 500,000 people. You're going to have to pick individual communities and you really want to know where. And so we've been asked to take sort of sub-sewershed samples, in which we've done. If there's not pre-existing sub-sewershed sampling points, which sometimes exist, but not very often, it's a lot harder. I knew it was going to be hard and complicated and difficult. It was a lot harder and more complicated and more difficult and more expensive than I thought that was going to be, because it gets down to picking a manhole, dropping an auto sampler down there, couple, 3 times a week, and that gets hard for everybody involved, the facility included. And they've been great partners and been willing to try to do that when they can, but it's not something really we can ask of them on a long-term basis. So you got to find a balance there as well. And I think the last part there, how's it influenced or modified efforts? A lot of it has been that allocation of clinical testing resources that I was talking about a little bit before, to try to find the cases that are contributing the viral signal that you're ultimately detecting in the sewershed. Because if you find those cases, then they just go through the normal case investigation and quarantine process. You don't necessarily have to do anything special there. So that's been a lot of the response to the data.
Great. And so as we wrap up the webinar, I quickly want to share a couple of other points. This webinar is part of the Internet of Waters peer-to-peer network, and you can see we have folks represented in many states. However, if your state is white in this picture, that means there's no one from your state. So please consider joining the peer-to-peer network by going to the Internet of Waters website. Under the Connect Us, you'll see the peer-to-peer network. We also want to pause for a moment and let you know that this conversation will be continued at the peer-to-peer forum, which you can see at this link right here. And so if we didn't get a chance to get to your question here in the webinar today, you can post your question in the public channel there and our speakers will respond to you. And then we also have a quick poll, if you'll please participate in that poll. I'm going to give it a moment here. And otherwise, just to reiterate, we will post the video of this webinar along with the presenter's slides on our website. Lily's giving me a message. We actually have one other additional poll. So if you'll just hang on just a moment longer and let us get to both of those. But we plan to post the video of the webinar in addition to the slides on our website. And if you register for this webinar, you will also receive that link via email. And again, we hope that you'll consider joining the peer-to-peer network. We have this series of webinars, but we also have another exciting webinar coming up in January on North Carolina's Project Nighthawk, which is a drought communications project from the North Carolina State Climate Office. So that's on January the 28th at 3 p.m. Eastern Time. So Lily, we have another poll, or is this both of them? Oh, and that's like, here's the second one. So if you'll please take a moment and participate in this second poll, we very much appreciate it. I thank you so much, Greg, Nathan, Joe, for speaking today. We appreciate your work and efforts in this realm and your participation in the Peer-to-Peer Networks webinar series. I see there's one more thing in the chat room. Greg has placed in the chat room for all of you more California COVID sewer shed information, a link for that. So please take a moment and go visit that. And thank you again so much for attending. We very much appreciate it.